Welcome back, everyone. Today, we've got a really fantastic explainer lined up for you. We're going to walk through a fascinating scientific poster that digs into a normally harmless gut bacterium called Enterococcus faecalis. Now, here's the catch. Under the right conditions, this little guy can turn into a pretty serious opportunistic pathogen. We're looking at a study done right in the heart of a densely populated slum community in Mymensingh, Bangladesh, just to see exactly how this pathogen is behaving out there in the real world. Here's our quick roadmap for today's guided tour. We'll start with the One Health context, unpack some genetic clues, map out the antibiotic resistance, look at the rather starry resistance biofilm link, and then wrap up with the preliminary findings and next steps. All right, part 1, the One Health context. Let's set the scene. To really get a grip on antimicrobial resistance, or AMR, we have to look way beyond the hospital walls. The researchers here applied what's known as a One Health lens. Basically, this is an integrated approach that recognizes human health is intimately, literally tied to the health of animals and the environment we all share. So instead of just swabbing humans, the researchers collected 100 diverse samples from a single, interconnected slum community. We're talking 33 from humans, 33 from animals, and 34 from the surrounding environment. And that leads us to the big, central question driving this entire explainer. Are these resistant, virulent, biofilm-forming strains of E. faecalis neatly contained in one isolated group? Or are they just freely circulating and transmitting between the people, the animals, and the environment they share? Let's dive into part 2, unpacking the gene data. Out of those 100 samples they collected, 33 tested positive for E. faecalis. From there, the researchers randomly selected 21 of those isolates, so exactly 7 from each sector, for a really in-depth genetic breakdown. Taking a look at this bar chart showing virulence and resistance genes, notice the left side. 3 genes, gel E, spray E, and pill, were found in almost every single isolate, sitting right at over 85% occurrence, entirely regardless of where the sample came from. What that tells us is that these bacteria essentially share a common core virulence toolkit, no matter where they live. But it's not perfectly uniform. While there's a baseline of virulence everywhere, the human samples actually carry some extra baggage. For example, the SIL gene, which produces a really nasty toxin called cytolysin, is almost exclusively found in the human isolates and is practically absent in the animals. Now, check out this Pearson correlation heat map. Instead of just looking at how often genes show up overall, this shows us which genes actually travel together inside the very same bacteria. I love how the poster presenter described this. It's literally a guilt by association map for genes. If 2 genes are always hanging out together, their intersection lights up deep red. Notice that perfect 1.00 correlation between the gel E and spar E genes? That makes total biological sense because they sit in the exact same operon, meaning they get inherited as a package deal. It's a really fantastic internal check that proves this data perfectly aligns with real-world biology. And this next chart brilliantly illustrates those gene combinations numerically. You're looking at an upset plot. Look, if you've ever tried to decipher a Venn diagram with 9 different intersecting circles, you know it's a visual nightmare. This chart is way cleaner. It lists every unique gene package found in the isolates and shows exactly how many isolates from each specific source carry that exact package. The takeaways here are pretty striking. The tallest bars, which represent the most common gene packages, are just that core virulent set without any resistance genes, and those span all 3 sources. But when we look at combinations that include the BLA SHV or BLA TEM beta-lactamase resistance genes, they're rare, and when they do pop up, they are heavily associated with the human-derived samples. So humans represent the much higher risk subgroup here. Let's move on to part 3, mapping antibiotic resistance. The fingerprints. Now that we know their genetic potential, we need to pivot and look at their actual physical behavior, the phenotype. But before we look at the charts, we have to call out a vital discrepancy in the data. Genotypically, in the DNA, the researchers only detected those BLASH-SV and BLATAM resistance genes. They completely missed the standard VAN-A gene. However, phenotypically, they observed actual, physical resistance to the antibiotic vancomycin. This genotype-phenotype gap is a huge finding. It strongly suggests there are alternate resistance mechanisms happening here that the standard gene panel just didn't catch. Now, what's really fascinating about this antibiogram heat map is that it's essentially the true heart of the poster. Think of it as a report card for every single bacterial sample. Red means it resisted the antibiotic, teal means it was susceptible. If resistance was source-specific, like, say, if animals had their own completely distinct resistant strains, we'd expect the color-coded source bars on the left to clump together into solid blocks of orange, blue, or green. But look at that massive, highly resistant red block sitting right at the top. The sources are totally mixed. Human, animal, and environmental isolates are sitting right next to each other, behaving identically. And if you want statistical proof to back up that heat map, here's the PCA biplot. Each dot is a sample, positioned by its overall resistance profile. Those dashed circles are the confidence ellipses for the animal, human, and environmental sectors. See how much they overlap? This is direct statistical evidence that resistance phenotypes are completely shared right across the human-animal-environment interface. They absolutely do not care where they came from.
Which brings us to part 4, the resistance biofilm link, the smoking gun.
So the crucial point so far is that resistance is shared. But to understand the true public health threat, we've got to layer on the biofilm data. Follow the flow of the striking Sankey diagram from left to right. It tracks the path of the isolates from their original source into whether they are multidrug-resistant, meaning resistant to 3 or more antibiotic classes, and finally, into how strong of a biofilm they can actually form. Notice how heavily isolates from all 3 sources flow right into the multidrug-resistant, or MDR, category in the middle. And from there, the thickest, most prominent bands lead straight into the strong or intermediate biofilm formation categories on the right. Guys, this is the nightmare combo: multi-drug resistance paired with strong biofilms. This matters immensely because biofilms act like a physical shield. They literally protect the bacteria from antibiotics and from our immune system. It makes infections incredibly difficult to treat and allows the bacteria to just persist for long periods on surfaces or in the shared environment. These strains aren't just resistant, they are biologically primed to survive and stick around. Finally, part 5, preliminary findings and our final verdict. Let's bring all these clues together. The overarching take-home message is super clear. Human, animal, and environmental strains look genetically and phenotypically similar. When you have a bacterium showing a massive 93.3% resistance rate to nitrofurantoin and it's circulating freely through animals and the shared environment, you simply can't just monitor local hospitals. It's a textbook example of why a unified One Health approach to antimicrobial resistance surveillance is absolutely critical. Now, I do want to emphasize, just as the researchers did, that this is an early-stage preliminary study. It's totally the first of its kind for this community, and we're really just getting started. It obviously comes with limitations, most notably the really small sample size of just 21 in-depth isolates. To actually confirm true transmission pathways, rather than just relying on phenotypic and gene marker associations, the next step is to scale this up considerably. We're talking whole genome sequencing and metagenomic-based approaches. They're also planning to deeply investigate that totally unexplained vancomycin resistance gap we talked about earlier. So to be clear, these are preliminary findings from an ongoing study, and there's still a long way to go. But even at this early stage, the pattern is undeniable, and frankly, a bit alarming. It's a massive first step toward understanding the true dynamics of AMR at the human-animal environment interface. I'm happy to answer any questions you have, so drop them down in the comments. Leaving you with a thought for the day: how will the One Health approach change the way you view the spread of superbugs? Keep learning, stay curious, and I'll catch you on the next explainer.